So we are going to begin talking about photosynthesis and cellular respiration. And when we look at it, it's going to be much more in depth than when you did, when you studied this freshman year, because we're going to look at all of the biomolecular phases that happen during those reactions. Freshman year, you just knew the big process. You know that plants use water and carbon dioxide, and then light happens and magic, you have sugar. We're going to actually go through those steps on how we turn carbon dioxide and water into sugar using light energy in a leaf. Part of that is a series of reactions that we call oxidation reduction reactions. I have no idea if you guys got to this in chemistry last year. Did you? A little bit? Okay, so some people have heard oxidation reduction reactions before if you were in honors chem. If you're in regular chem, it's not even in there at all. And some of you are in chem now. The good news is that you don't need to know a lot about oxidation reduction reactions in bio. You just really need to know what oxidation means and reduced, because we'll use those words a lot. So I remember oxidation reduction by that acronym oil rig. So oil, O-I-L, oxidation means it's losing. And what we're talking about in that reaction is electrons. So if something is oxidized, it lost electrons. And we're going to use the word oxidized a lot. The opposite of oxidation is going to be reduction, or I'll say the word reduced a lot. Rig, an oil rig, stands for reduction is gaining. So that means something gained an electron. So if we look at this simple little reaction here where we have sodium and chloride and they come together to become sodium chloride, we know that sodium loses an electron. So that is oxidized. Anything that loses an electron is oxidized. That electron has to go somewhere. It went to chlorine, and we see that negative charge right there, so we know that that electron went to chlorine. So chlorine was reduced. We are not working with ions in these reactions. So these metabolic reactions aren't going to have pluses and minuses, so where we can track the electrons, but we need to know where the electrons are going, because the movement of electrons, that's the power that allows us to do these reactions. So the movement of electrons is super important in photosynthesis, but also in cellular respiration. So we need to kind of track where those electrons go. There's no charges here because these are all covalent bonds, but electrons are negative, hydrogens or protons are positive. We know where the electrons go if we track the hydrogens. So if we follow where the hydrogens go, that's where the electrons are. So if we look at this reaction, this is methane. It's a very simple organic molecule, 1 carbon, 4 hydrogens. We're going to track where these hydrogens are going, and they're leaving this carbon, but they're going on to this oxygen to form water. Because see over here how oxygen now has hydrogens and it didn't? So if we're talking about this reaction, we're going to say that methane is oxidized and oxygen is reduced. So we just track the hydrogens, and we pretty much just track the hydrogens on the reactant side. This had hydrogens, this didn't, so this was oxidized, this is reduced. So we have 2 reactions here. These are big, huge metabolic pathways, and there's lots and lots of reactions that happen, but our overall process is we need to know what is oxidized, where do we get electrons from, and what is reduced, where do those electrons go. Here we have the chloroplast. There's chlorophyll pigments as well. Those are like the green pigments, as well as other carotenoids, which are like the yellows and the reds and the oranges that we see now, because plants aren't still making chlorophyll, but so we can see their carotenoids, but not their chlorophyll. So that's why they look different colors. But we know that when light hits the chloroplast, that light energy is used to convert carbon dioxide and water into sugar. And then its waste is going to be oxygen. So we're taking light energy and we're storing it as chemical energy here. When we look down here at the opposite of photosynthesis, that's cellular respiration. And remember, both plants and animals and fungus can all do cellular respiration. Sometimes we think plants don't do cellular respiration, but they do. Here we have sugar combined with oxygen, and then we are making carbon dioxide, water, and then here are energy-rich molecules, ATP, that we need to do cellular work. So we're converting one form of chemical energy into a new form of usable chemical energy that can do work. Tracking where those hydrogens are and where they go. In the chat, tell me what is oxidized and what is reduced in photosynthesis. Oxidized is losing, reduced is gaining. Okay, so I see a bunch of different answers, some correct, some not. I'm not surprised because this is a lot going on. Notice here how water has the hydrogens. Carbon here does not, carbon dioxide does not. So water loses these hydrogens. If you're losing hydrogens, you're also losing electrons. So water is being oxidized. Electrons are going to be stripped from water. And that's why plants need water. They need the electrons from water to do photosynthesis. So water is oxidized. Where do those electrons go? Well, this carbon had no hydrogens, and then over here there's a bunch. So carbon dioxide is reduced. So in photosynthesis, water is oxidized, carbon dioxide is reduced. What's oxidized and what's reduced in cellular respiration? Go ahead and put that in the chat. Much, much better, those of you who are responding. We know that sugar or glucose is being oxidized. Electrons are taken off of sugar and they're put on to oxygen. So oxygen is reduced. So oxygen is reduced to form water. The sugar is oxidized. Good. If you are still kind of struggling with these words, we're going to say them over and over and over and over again so that they're going to become very familiar. Just remember that when something is oxidized, it loses electrons. When something is reduced, it gains electrons. But I really just want you to be familiar with those two terms. Let's use our annotation. Let's just put a stamp on what is oxidized. Very good. So I see most stamps right here on these 2H. This is being oxidized. I know that because now it's positive, it lost its electrons. All right, a stamp on what is reduced. Good. NAD plus is reduced to form NADH. And we can see, we track where those hydrogens are going. That gained an electron, so this was reduced to form NADH. We're going to see NADH a lot in cellular respiration and in photosynthesis. This is an electron carrier. Okay, how about a stamp on what is oxidized? Nicely done. This one's kind of nice because it's showing you where those electrons are because it's making the hydrogens that are by those electrons pink. So we have this organic molecule that is being oxidized. What is reduced? Great. So FAD is reduced to form FADH2. So FAD is reduced to form FADH2. This is also an electron carrier that we are going to see in photosynthesis. So understanding those electrons and where they're moving, that was our main purpose for the day. You guys are doing much, much, much better at it. Okay, so our final thing for today. Here is our chloroplast that we saw before. And we talked about this when we talked about endosymbiotic theory because we said chloroplasts had their own ribosomes, they had an inner and an outer membrane right here, and they had their own DNA. And those were all pieces of evidence that supported that chloroplasts came from ancient prokaryotes. What we are going to add to that today is some more features of this chloroplast. So this almost looks like a poker chip, right? And they're all stacked on top of one another. The one poker chip right here is called a thylakoid. And then notice that the stack of poker chips here, and you can see it on the electron micrograph here as well, that's called granum, or that's a plural, granum. And notice that they're green, right? So here in the thylakoid membrane are lots of pigment molecules and a lot of chlorophyll in the thylakoid membrane. And then the other thing that we have out here, this stroma, this is just the liquid, the fluid-filled liquid that's in the chloroplast. And that's the only new thing that we're going to talk about today. Lower, and I'm going to record this as well so you can watch it later. So let's first just talk about the chloroplast structure. So many times we think of photosynthesis happening just in plants because plants do have chloroplasts, but the reactions that we're going to talk about also happen in things like algae, which are single cellular, and they also happen in like cyanobacteria. So some bacteria can do photosynthesis as well. Now, the bacteria obviously don't have a chloroplast, but they carry out these reactions in their cell membranes instead of sequestering it into just the chloroplast. But things that we had talked about before, there's two membranes of the chloroplast. There's this outer membrane, and there's the inner membrane. And that's one piece of evidence that supports endosymbiont theory, the theory that plants and chloroplasts specifically evolved from ancient prokaryotic photosynthetic organisms similar to like the current cyanobacteria. So that we knew already. Some things that we talked about like just briefly before, this one tiny green circular thing, just one of them, that is called the thylakoid. And the inner part of that thylakoid is called the lumen, or sometimes your book refers to it as the thylakoid space. It's that space inside the thylakoid. And they're flat green discs with like an aqueous solution in the inside. A stack of these all together, so this stack of thylakoids, we refer to that, if it's singular, as grana. But if we wanted to talk to them, like if there's multiple grana here, the plural form of that word would be granum. So one would be grana, two or more granum. And all that is just stacks of thylakoids. And I think of a thylakoid like a poker chip, and when you have stacks of poker chips, that would be like a grana. The other thing that we have here is there's fluid surrounding the granum, and this fluid is called the stroma. We're going to focus a lot on the thylakoids today because notice the thylakoids are green. The thylakoids have the pigment molecules like chlorophyll A and chlorophyll B. They also have other pigments, the carotenoids, that have colors that are like yellow and orange and reds. Normally, we don't see those colors in plants unless it's fall, like it is now, because when it's fall, plants stop making chlorophyll. Chlorophyll is a molecule that they have to consistently make. When they stop making chlorophyll, then we see those other pigment molecules, those carotenoids that are in the reds and the oranges and other colors. So here's our structure of the chloroplast. Now we're going to look at a plant and kind of look at just leaf anatomy in general. So notice here that we're looking at some of the inputs that are necessary for photosynthesis and how they enter the entire organism. We see that water is a necessary input of photosynthesis. That's going to be absorbed from the roots. It's going to travel up the xylem. We don't have to worry about what xylem kind of is now, but it's kind of like hollow cells that act like a straw. And the water travels up the xylem and comes into the leaf. And the leaf is the organ that's going to carry out photosynthesis. So we need to get water into the leaf. It comes up through the roots, is absorbed, and it comes up through the plant, through the xylem cells. The other input that we have here is carbon dioxide. Carbon dioxide is a gas. It's in the atmosphere. It also needs to enter the leaf. It's not coming up from the roots, but instead, if I took a leaf And this here is a cross section or a longitudinal section of a leaf, and it's magnified. So if I had a leaf and I cut it and then I magnified it to look and see what was inside the leaf. Here on the top of the leaf, we call that the epidermis. Just like our skin is called the epidermis. So it's like the outer covering. So there's epidermis on the top and the bottom of the leaf. And then you can see these green cells inside, and those green cells, we don't really care what they are, but these are the mesophyll cells. These are the ones that have chloroplasts. And we can see that because they're green. We can see that pigment molecule chlorophyll. But what I want to show you here is that there are little pores here, little openings. And usually they're on the underside of the leaf. Under the leaf, if we look at it under the microscope, we see these openings. These openings, one opening is called a stoma. Multiple openings, the plural form, is called stomata. And they're just holes in the bottom of that leaf. Here, we have microscopic images of a stoma that is closed and a stoma that is open. So the opening right here, this is the stoma. It's just the opening. The cells on either side of the stoma are called guard cells. Kind of like if you have, if you're coming into a castle and you have the opening of the castle, you have guards on either side. That opening is a stoma, and then you have cells on either side of that opening. And those are guard cells. Now, these guard cells are going to be very important. They're very important in maintaining the homeostasis of the plant because the stoma needs to be open so that carbon dioxide can enter the plant. And we know that carbon dioxide is needed for photosynthesis. It's one of our inputs. But I want you to see something else. Notice here that water can evaporate and come out of the stoma. When that happens, that process of water leaving the stoma, that's called transpiration. Kind of sounds like perspiration, right? Like when you sweat and you perspire, water comes up on your epidermis, and when it evaporates, we get evaporative cooling. It keeps us cool. The same thing happens with plants. They don't have sweat glands like we do, but as soon as their guard cells open the stoma, carbon dioxide comes in, but water leaves as well through evaporation, which is not bad if that plant is full of water. But remember, water is necessary for photosynthesis. If plants lose too much water, they can't carry out photosynthesis any longer. So they always have to maintain a balance. We need water. If we lose too much water, close the stoma. But we also have another balance where we need to do photosynthesis. And if I don't have carbon dioxide, I can't do it any longer. So open the stoma to bring in carbon dioxide. So the guard cells have to maintain that balance of, staying open so that we can bring in carbon dioxide, but being shut at other times so we don't lose too much water through transpiration. And then the last thing that's important for the stoma is this oxygen. That's an output or a waste product of photosynthesis. And that waste or that oxygen needs to leave. So the underside of the leaf doesn't get a lot of attention, but it is really important in maintaining the homeostasis of the plant. It makes sure that the plant has enough carbon dioxide to carry out photosynthesis, and then it helps regulate how much water will be lost through transpiration as well. And it's another port for oxygen to leave the plant after photosynthesis is done. These guard cells are not smart. Like they don't have conscious thought. They don't have they're not getting data on how much water and carbon dioxide the plant has. Instead, though, these guard cells use osmosis and how much water the leaf has to regulate whether they're opened or shut. So notice here that these guard cells, when they're open, look how fat they are. They're fat and they're full, and that word in a plant is called turgid, filled with water. So when they're in an environment that is very hypotonic, meaning there's lots of water surrounding them, water is going to rush into the guard cells and they're going to become turgid because they're filled with water. And if those guard cells are filled with water, they open the stoma because they get really fat and they open up. When the plant loses too much water, the first place where they lose water from are these guard cells. So if too much water is lost, these cells become flaccid. And when they become flaccid, those cells flatten and they close the stoma. So if water is plentiful, guard cells are opened up, carbon dioxide can come in. If environmental conditions change and there's not a lot of water and plants are losing a lot of water through transpiration, those guard cells close so that water can be conserved. I want to make sure there aren't any questions before we move out of like this gross anatomy. Okay. So let's move on to page 2. We're going to do 2 things. We're going to look at the light reaction twice because there's a lot going on. We're going to first like step back and just like see the big picture on what's going on. And then when we understand the whole process, we're going to look at it more closely. What I want to show you down here is that I'm going to talk about a lot of stuff. I'm going to use a lot of words. Okay. And as I go through that, it's hard to write everything in your notes and pay attention to what I'm thinking about and kind of understand the process. So instead, as I go through this, you can label this diagram when I talk about each of these processes. So you can just label those instead of writing everything on the diagram that happens. So let's first just look at this model and understand what we're looking at. Notice here that this says inner thylakoid. Sometimes we also call that the lumen, the inside space of that poker chip. And if this is the inner thylakoid, this right here is the thylakoid membrane. And it's showing it like a U, but understand that this thylakoid is a circle and that U would come all the way around, but we're not looking at everything. We're only looking at half of the thylakoid, half of that U. Notice that it's made of phospholipids. Remember that the inside is hydrophobic because it's made of fatty acid hydrocarbon tails, but we have charged phospholipid heads. The other thing is if this is one thylakoid, surrounding the thylakoid is the stroma. So if we look back where we are looking at one thylakoid right here. The inner part right here is the lumen. Here's the membrane, and the stroma is the fluid that surrounds it. So if you know where we are, thumbs up for me. Okay. So we're just talking about the light dependent reaction. Photosynthesis is broken up into two two like major reactions, the one that requires light and the one that does not, or the light independent reaction. This one, light is actually like necessary. So if we look here at A, it says light excites electrons to move to the electron transport chain. That's happening right here. Here's light coming from the sun. What it is hitting here, this is called photosystem 2. What the photosystem is, it's a group of proteins that hold a whole bunch of pigment molecules. And a pigment molecule is just a molecule that we can see because it has a color, but those are molecules that can absorb light. And if light is absorbed, we can harness that energy from the light and they hold on to it. And what they do is notice that each of these right here, these are all pigment molecules. And they act like a satellite dish. And a satellite dish takes in all of that information, all of that light energy, and it funnels it to one place down here. And the pigment molecule that it funnels it to is going to be chlorophyll A. Now, there's a whole bunch of pigment molecules. There's chlorophyll A, there's chlorophyll B, there's carotenoids, there's a whole bunch of them. But all of that energy is harnessed by all of those pigment molecules, and it goes down here to chlorophyll A. When all of that energy hits the chlorophyll A and is focused on that molecule, what happens is B right here. This is an electron. And it's so excited because it got so much energy from the sun that was focused on it from all the other pigment molecules that that electron gets excited and it leaves the molecule. It travels from photosystem A to proteins in this electron transport chain. ETC stands for electron transport chain. It is a chain of proteins and they pass the electron from one protein to the next, and it just moves down the electron transport chain. So remember in chemistry when you guys had like your first unit and you talked about the flame test? And in the flame test, you took some of the inorganic like salts and you put it under the Bunsen burner and we saw a beautiful like color change. What happened was you were exciting the electron and the electron moved up, and then when it fell back down, it released that energy that we saw as light. These electrons are getting excited just like they did in the flame test, but more so because when you did the flame test, those electrons got excited, and then when they fell back down, we saw light. This is not happening. This electron's getting so excited that it leaves the molecule completely. So this portion is adding so much more energy than even that Bunsen burner because the electrons are getting more excited than they did in the flame test because the electrons actually leave. So now we have chlorophyll A. It has lost an electron. So it has like an electron hole right here. It's lost it because it's moved out of photosystem 2 and it's traveled down the electron transport chain. If we're in a good spot, thumbs up. Okay. All right, so now we're ready to come here to step C. And notice that light is happening again. You know what, before we get to C, we just talked about the electrons moving down the electron transport chain. Let's come back here to B. Movement of electrons is electricity, right? So that's what we use, like if I plug something into the outlet, power happens because of the movement or the current of electrons. So this is moving electrons. This is an energy source. That movement of electrons is going to power proton pumps in the electron transport chain. So see how I have a hydrogen ion here, and it's moving from a low concentration of hydrogen ions to a high concentration of hydrogen ions. That is active transport. That needs energy. That energy is not coming from ATP, it's coming from the movement of electrons down the electron transport chain. It's coming from that current of electrons. So proton pumps are powered by the movement of electrons through the electron transport chain, and now I have this high concentration of protons. Now we're ready for C. Here's my electron. It's down at the bottom of photosystem 1. This is a stupid thing that biologists have done. This is a photosystem I talk about first, but I call it photosystem 2. And then after that comes photosystem 1. They're out of sequence. It's stupid. It's dumb. But photosystem 1 was the first photosystem that was discovered by scientists. And then later on, they discovered photosystem 2. They just saw, oh, there's 2 photosystems. They did not understand how they work together. So because they found this one first, they called it photosystem 1. Because they found this one second, they called it photosystem 2. But we know that photosystem 2 comes before photosystem 1. And it's stupid that they didn't rename them. It's dumb. So sometimes I like to call this the P680 photosystem, and then I call this photosystem the P700. Because 680 comes before 700. Sometimes AP will call it that, sometimes they'll call it photosystem 2. The reason that we give them these numbers is because this is the wavelength that photosystem 2 absorbs light best at, and this is the wavelength that photosystem 1 absorbs light the best at. It's stupid how they're named, but it's based on when they were discovered. So, step A, light hits photosystem 2, harnesses all its energy onto chlorophyll A, the electron gets excited, and it moves down the electron transport chain, which powers the proton pumps to fill up the lumen with a whole bunch of protons. Then the electron is now at the bottom of photosystem 1 in another chlorophyll A molecule. Light hits photosystem 1. It excites that electron again, so much that it leaves chlorophyll A, and eventually an enzyme is going to catch that electron, and that enzyme is going to put that electron onto NADP+. So this is NADP+. Has a positive charge. When that electron is taken by NADP+, it is reduced to form NADPH. I don't see that electron anymore, but I see the hydrogen, and I know the electron is there because hydrogens follow electrons around. This is our electron carrier. That electron carrier is going to be really important because we're going to need that electron later when we go on to the Calvin cycle. But that is an energy-rich molecule carrying an electron for us, and it's going to go on to the Calvin cycle. Notice here that I have an electron that's lost from chlorophyll A in photosystem 2, goes down the electron transport chain, gets excited again, and then now it is on NADPH. This chlorophyll A right here, I'm going to talk about step E right here. That chlorophyll A has a hole in its heart. It lost its electron. It's gone. That electron needs to be refilled, or this process can't happen again, because this is not the electron comes back around. No, it's not cyclical. It's linear electron flow, because this electron's here. It is lost. It's going on to the Calvin cycle. This electron needs to be replenished, and here's water That is one of our inputs of photosynthesis, right? We can't do photosynthesis without water. What water is, it's an electron donor. It's going to be, water's going to be oxidized. It's going to lose its electrons, and we're going to form oxygen gas. And this is our output or our product of photosynthesis. So here's our reactant, here's our product, and what water is doing is it's donating its electrons to chlorophyll A, and it's filling that hole that chlorophyll A had. What we need to talk about right here is D. This is ATP synthase. I know it's an enzyme because it ends in ASE. The movement of electrons created this high concentration of protons in the lumen or in the inner thylakoid space. This means that I have a very low pH. This is a very acidic environment. Lots of protons. Out here in the stroma, I have a low concentration of hydrogen ions. It is very basic, and it has a high pH. These protons will move through ATP synthase, through this enzyme. That movement does not require any energy because we're moving from a high concentration to a low concentration. So it's moving by facilitated diffusion. But the movement of these protons through ATP synthase is going to, like, it literally, as those move, like there is a wheel inside that enzyme, and that wheel, as it turns, and as these protons move through ATP synthase, they take ADP, they add phosphate, that PI stands for inorganic phosphate, and it attaches the phosphate onto ADP, so it phosphorylates ADP to make ATP. And this is our energy-rich molecule. ADP will also move on to the Calvin cycle. So the whole purpose of this light reaction is to make electron carrier, NADPH, and to make ATP. It takes energy from the sun to move electrons, power proton pumps, and create our energy-rich molecules that are needed for the next part of photosynthesis, which is called the Calvin cycle. I want 3 questions because we talked about a lot. So either, like, I know that there's questions, so I'm not going to move on until I answer a couple of them.
Okay, so when you say that they move on to the Calvin cycle, do they both? Because like that's just the part that I'm kind of confused on, like what happens after that.
So we are going to talk about what happens after that tomorrow. But yes, NADPH, our electron carrier, and ATP, our energy-rich molecule, both go on to the Calvin cycle. But the Calvin cycle happens in the stroma. And if you look at your model, notice that ATP and NADPH are out of that thylakoid and they're in the stroma now. So they're ready and they're out in the stroma, so they go right into the Calvin cycle.
Okay, thank you.
That was a good question. 2 more.
So step C, basically when the electron turns into the NADPH.
Mm-hmm.
Okay.
So I would say in step C, the electron in chlorophyll A is excited. I'm going to go through it in a little more detail when we go over the next model, but that electron eventually is transferred onto NADP+ by an enzyme.
So the actual like transfer of electrons comes from water breaking down, like the electron coming from water, and then the light ends up actually like making that reaction happen.
Mm-hmm. So one of the most important things here today is to know the role of water in photosynthesis. So water acts as an electron donor. Sometimes they call this the water splitting reaction because we need to break water and we oxidize it. We remove electrons from water and those are transferred onto chlorophyll A. Eventually, after light hits those photosystems and the electrons move, eventually they're going to end up on NADPH, on that electron carrier. But without water, none of this could happen. One other thing that students struggle with a lot is understanding pH and how that relates to hydrogen ion concentration. So I'm not sure what you guys put into your notes, or if you are writing it in long form, or if you're using Cami, or if you're going to watch this later on and then take notes. But one thing that many students struggle with is many times AP will ask you about the inner thylakoid spacer, the lumen, and they'll say that it has a low pH, which is true. But a low pH means that it has a high concentration of hydrogen ions, which I know you guys know, but there's so much going on that when we have those indirect relationships in our mind, those are hard for our mind to handle sometimes when we're overwhelmed with information. So make sure that you know that a high concentration of hydrogen ions means that that has a low or acidic pH, and that the stroma, because it has a low concentration of hydrogen ions, means that the pH is high. That is one thing that students always struggle with every year. We also know that these proton pumps, this is active transport, and that this energy source is coming from the movement of electrons, but the movement of the protons out into the stroma, that is facilitated diffusion. There's no energy needed for that because they're moving from a high concentration to a low concentration. Anything? So all of these protein complexes are the electron transport chain. The second complex right here, photosystem 1, or sometimes we call it the P700 photosystem because it absorbs light well at 700 nanometers. Finally here, these are some things that really weren't on our last model, but this is an enzyme, and this is called NADP+ reductase. And it ends in ASE, which is an indicator that that's an enzyme. Down here is ATP synthase. It also ends in ASE, which lets us know that that is also an enzyme. And remember, enzymes are proteins that catalyze reactions. Catalyze means to speed up. So now we have all of the structures pretty much labeled on that are embedded in the thylakoid membrane. And now we can kind of go through the process of the light reaction. One thing that I want to make sure that you understand is the linear flow of electrons. Because we're going to talk about the Kelvin cycle that happens after the light reactions, and that's a cycle. It like just goes around and around and it regenerates. But this is not a cycle. This is a linear flow of electrons. So we're going to start here with photosystem 2, and we know that light from the sun is going to hit that photosystem, and all of these green little dots embedded in it represent pigment molecules. There is chlorophyll A, there's chlorophyll B, there's carotenoids that aren't green but other colors, but there's lots of pigment molecules, and they all focus that energy from the light onto these darker green molecules, and this is a chlorophyll A molecule. When all of that light energy is focused onto chlorophyll A, it excites an electron away from the chlorophyll A molecule so much that that electron leaves the photosystem and it proceeds down the electron transport chain until it rests on the chlorophyll A molecule of photosystem 1. So we have this electron flowing down the electron transport chain and moving from photosystem 2 to photosystem 1. That current, that movement of electrons is going to power proton pumps, and it's going to pump protons. So in the electron transport chain are proton pumps, and they're going to move hydrogen ions from a low concentration in here where they are at a high concentration. And it is the movement of those electrons that allows that active transport to occur, because we're going from a low concentration in the stroma to a high concentration in the lumen. Because of this, because of the pumping in of these protons, we know that this is going to have a high concentration of hydrogen ions, and that means that we have a low pH, and that the lumen is very acidic, because these protons cannot move across the phospholipids of the thylakoid membrane because they're ions and they have a charge, and charged particles cannot pass through the phospholipid fatty acid tails that are hydrocarbons and hydrophobic. They're sequestered in the lumen. Light will hit photosystem 1. All those pigment molecules will absorb that light. They will transfer that energy to the chlorophyll A molecule of photosystem 1, excite that electron, so it will leave that photosystem. It will be transferred through other electron transport proteins until it gets here to this enzyme NADP+ reductase. That NADP+ reductase is an enzyme that will transfer that electron onto NADP+ to form or to reduce NADP+ to form NADPH, and this is our electron carrier. So we said that this is linear electron flow, meaning that the chlorophyll A molecule lost its electron from the P680 photosystem. That electron has moved through the electron transport chain to photosystem 1, and now eventually it ends up on this NADPH electron carrier. That's where that electron is, right there. So we've made one of our energy-rich molecules our electron carrier. The other high energy molecule that we need to make is ATP. So here's ATP synthase. This is an enzyme that synthesizes or makes ATP. So the movement of protons through ATP synthase is going to provide the energy to create ATP. This ATP is our energy-rich molecule that is needed to perform coupling reactions. Because remember, we said that anabolic reactions are not spontaneous and they're not going to happen unless they're coupled with the dephosphorylation of ATP. And we're going to try to build sugar molecules to build bigger molecules, and that's not going to happen on its own unless we have ATP to couple those reactions with it. So we have our electron carrier, we have ATP, and they're going to go to the Kelvin cycle that is out here in the stroma. Sometimes we also call that the light independent reaction.
Ms. Baxter,
Yep,
can you slow down just a little bit?
of course, yeah.
Thank you.
You're welcome.
And one more question. I can't see what it says. It's like a word in the middle of like whatever that cell or whatever, right below like the P680, right, and to the right a little, and down.
Proton pump?
No, right below it, inside of it.
ETC?
No, right below that.
Okay, so I have an arrow pointing here to chlorophyll A, right here. And then I just have electrons moving down the electron transport chain.
Oh, okay. Thank you.
You're welcome. How about any other questions? Because we've just reviewed the light reaction one more time. Okay, so let's see how you guys are feeling. I know yesterday you gave me like your fingers and you were telling me how you were feeling, and we've reviewed it one more time. How confident do you feel with the light reaction now that we've reviewed it a second time? 5 is good, 3, 1 is bad. Okay, great. I think everyone that I can see like feel better today than they did yesterday. So let's move on to that Kelvin cycle. The light reaction is happening in the thylakoid, but the Kelvin cycle is going to happen in the stroma out here. What I want you to see is that NADPH, the electron carrier, and ATP are already in the stroma. They're already outside of the thylakoid. And that this is where the Kelvin cycle happens, is out here in the stroma already. So in your textbook, this is chapter 8, section 3. If you're going to review it, I will tell you that if you read that, there's a lot of information that you do not need for the AP test. They go through all of the reactions that happen here, and you do not need to know all of those reactions. So what I'm going to tell you is just what you need, not anything more. If you go back and review it in your textbook, that's wonderful. I just don't want you to be overwhelmed, because if I don't talk about it in class, it's not going to be on the AP exam. And for this section, they go into a lot of depth in the Kelvin cycle that's not on the AP exam. So we talked about some of the inputs into the light reaction. So some of those inputs was water, because water is needed to replace this hole in chlorophyll A where it's lost its electrons. So the first input of photosynthesis is water, and water is oxidized, meaning that the electrons are removed and they're put back onto chlorophyll A. So so that the light reaction can't continue. And then here's light, here's water. We know that those are two things necessary for photosynthesis. And then the last thing that we need for photosynthesis is carbon dioxide. But we don't use carbon dioxide until we get to the Kelvin cycle. So I'm just going to write the equation for photosynthesis up here. We're going to take water plus carbon dioxide, and then using light energy, we're going to make glucose. I'm going to just put 6's in front of there just to balance it out. This is needed in the light reaction. This is happening in the Kelvin cycle. We also have the product of oxygen gas, and we know that oxygen gas is produced up here in the light reaction as well. What we really need is we need to start making some sugar. So we're going to break the Kelvin cycle up into like 3 phases right here. And this is going to be phase 1, which will be carbon fixation. Phase 2 is going to be reduction. And phase 3 is going to be regeneration. There are going to be many, many enzyme catalyzed steps throughout this entire cycle. However, you just need to understand the major things that happen in each of those 3 phases, not every single step that your book talks about. So the first phase is carbon fixation. To fix something is a weird term that we use in biology, or we use it in environmental science. But when something is fixed, that means that we incorporate an inorganic molecule into an organic molecule. So organic molecules are molecules that are composed of carbon. We talk about organic molecules all the time in AP Bio because we talk about nucleic acids, we talk about proteins, we talk about lipids, and all of those have a carbon backbone, so they're organic molecules. The only molecule that has carbon in it that's not considered to be organic is carbon dioxide. Everything else that has carbon in it is considered to be an organic molecule. So carbon fixation means that we're taking carbon dioxide gas and we're incorporating it into an organic molecule, into a larger carbon-based molecule. We will also talk about fixation when we talk about the nitrogen cycle in Unit 8, a long way from now. But if you took AP Environmental, you know nitrogen fixation is when organisms take nitrogen gas from the air and they incorporate that into their organic molecules. So that's the only thing that fixation means. It's always going to take some inorganic molecule from the atmosphere and incorporate it into its own biological organic molecules. So that's our first step, is bringing carbon dioxide in from the atmosphere and incorporating it into an organic molecule. Remember that that carbon dioxide is coming into the plant through the bottom of the leaf, through the stoma. And that's how the carbon dioxide gets in there. Once the carbon dioxide is in the leaf, though, there's going to be an enzyme called Rubisco. Sounds like Nabisco crackers, but it's Rubisco. And it is that enzyme, Rubisco, that attaches carbon dioxide to RUBP. So we attach carbon dioxide to RUBP, and that is carbon fixation. And the enzyme that catalyzes that reaction is Rubisco. You do not need to memorize all of these things that I've talked about, but if they ask you questions about the Kelvin cycle, they may have things like this in the model, and you might need to interpret that model. So it's not something you need to memorize, but it's something you need to be familiar with. So when you see diagrams or you see models, you're not completely lost. So if you see this Rubisco biphosphate, RUBP, you can be calm and just know, oh, that's the thing that carbon dioxide attaches to, and the enzyme Rubisco does that reaction. It catalyzes that reaction. This molecule is important because RUBP is the only molecule that can accept carbon dioxide. So if we don't have RUBP, we can't fix carbon dioxide. And we can't incorporate it into organic molecules. And Rubisco is important because it's the only enzyme that can fix carbon dioxide to RUBP. It's probably the most abundant enzyme on the planet. So that's the end of phase 1. Put some carbon dioxide on RUBP and fix it. RUBP, it has 5 carbons. We attach the 1 carbon of carbon dioxide to it, and we get the 6 carbon intermediate that's very unstable, and then it quickly breaks apart into 2 pieces. Not a big deal that you know that, but if you read it in your book, it might get confusing. You don't need to know about all of that. But when we get to reduction, that is something that is important. So remember, we talked about oxidation and we talked about reduction. Oxidize means that you take electrons away. And we know in the light reaction that water was oxidized. We stole electrons away from water so that we had that linear electron flow. And those electrons ended up on our electron carrier NADPH. Now we're going to do reduction reactions. Now we have our electron carrier that's actually going to do something for us. Because if we look up here, we're trying to make sugar, right? We need matter to make that sugar. Here's the carbon that comes from carbon dioxide. Here's the oxygen that's coming from carbon dioxide. We need the electrons and the hydrogens. That's going to come from our electron carrier because here's NADPH. This has the hydrogens and those electrons that we need to make the sugar. That's where that matter's coming from, is that electron carrier. So in this reduction phase, we're going to use our ATP molecules. So they're going to dephosphorylate and become ADP. But then also our electron carrier is going to drop off its electrons and that hydrogen to those molecules as well. At the end of reduction, what we get, and we're not going to call it the PGAL because your book calls it G3P, and that's what AP calls it as well. It is the same name for the same molecule. It is just a 3-carbon sugar instead of a 6-carbon sugar. And that leaves the Kelvin cycle. So G3P is a 3-carbon sugar that leaves after the reduction phase. Glucose has 6 carbons. GP3 or D3P has 3 carbons. We put those 2 molecules together later on, and that is what makes glucose. But glucose is not made through the Calvin cycle. This 3-carbon sugar is made, and it's incorporated into other organic molecules like glucose. That's the end of phase 2. In phase 3, all we need to do is regenerate RUBP. That will require some energy coupling to do that. So it will require some ATP as well to regenerate that molecule. But then we just keep on going through the phases over and over again. Fix carbon dioxide, reduce it, add those electrons and hydrogens, have the 3-carbon sugar leave, regenerate RUBP. And it just happens over and over and over again. Notice though that this Calvin cycle, there is no light that's necessary for this to occur, but you need ATP and NADPH, or this will never happen. And these molecules are our end products from our light reaction. So if there's no light, we don't make the energy-rich molecules. And if we don't make the energy-rich molecules, we can't do carbon fixation and the Calvin cycle. How about 3 questions before we move on? You now know everything about photosynthesis. You have no questions at all? What I would like you to do is either in your notes, because we have about, we have till like 8:45. So I'm going to give you guys 5 minutes. What I would like you to do is the best that you can, let's organize the inputs and outputs of the light reaction in the Calvin cycle. Like what goes into the light reaction? Where does it happen? What are our outputs? What needs to go into the Calvin cycle? Where does the Calvin cycle happen? And what do we make at the end? Let's fill out that table. And then when you're done filling that table out, either on Cami or in your notes, just go ahead and let me know in the chat that you finished, and then we'll review it together. going to give you guys a little bit more time. I only have 5 people who are done, so it sounds like you guys need a little bit more time. If you are done and you haven't given me a chat, just go ahead and throw that in so I know you're finished. Okay, I know some of you are still working on it, but I want to make sure that we go over this together, and about half of you are done. So let's see. Megan, you finished. Which one of these boxes do you want to tell me how to fill out? You can choose any box you want.
I could just do the input for the light reaction.
Okay, what should I put in there?
So light and then water.
Good. So we need light. You should know that light is providing the energy that we need for photosynthesis. And then Megan, what is the purpose of water in photosynthesis? What does that provide? It's not providing energy, but what does water provide in?
Doesn't it does something for the chlorophyll?
Yes, yes. So anyone, give Megan some help. What does water provide to chlorophyll A in photosystem 2?
When it breaks down, it provides the electron.
Good. So water is going to provide the electron that's needed because we have linear electron flow, and photosystem 2 needs to have that electron replaced. And sometimes we call that the water splitting reaction because water is split apart, or we could say water is oxidized to remove those electrons. Jake, what box do you want to fill out?
Sorry, my thing is messed up. Give me 1 second.
No worries.
I'll do the output for the light reaction.
Okay, so what are our products of the light reaction?
NADPH and ATP.
Good. And then we know that NADPH, I'm going to underline this H, is because I'm not going to see electrons anymore, but I know wherever that hydrogen is, it's there because it followed an electron. So NADPH is our electron carrier. And ATP is our energy-rich molecule that we're going to need for coupling reactions. It's going to make non-spontaneous reactions possible, or anabolic reactions actually possible. Lainey, what box do you want?
I'll do the input for the Kelvin cycle.
Okay, what do we need in the Kelvin cycle?
I had carbon dioxide, ATP, NADPH, and then Rubisco.
You are absolutely right about all of those things. We know that carbon dioxide enters in through the stoma, the underside of the leaf. We know that these energy-rich molecules from the light reaction are needed. I'm just not going to write Rubisco down because it's an enzyme. And I want to make sure that you guys know that enzymes only catalyze reactions, they're not used. So Rubisco is part of the Kelvin cycle and it's very important, but I want to make sure that you know that enzymes are not used when they catalyze reactions. They just catalyze a reaction and one enzyme can keep that same reaction and catalyze millions of reactions. Anna, what box do you want?
I'll do the location of the light reaction,
Where's that happen at?
the thylakoid.
Okay. Leah, which box do you want?
I can do that output for the Kelvin cycle.
Okay, what do we make?
Okay, ADP and G3P.
So we make G3P. This is our 3-carbon sugar. We definitely made ADP because we dephosphorylated ATP. And then we took the electron and the hydrogen away from NADPH. So we also make NADP+. And then what I'm going to put here is just know that these are also needed for the light reaction too. Because we see that these outputs right here are going down here, and that we need to generate these for the light reaction as well. Ashton, where's the Kelvin cycle happen?
Doesn't it happen in the stroma?
It does. Great job, guys. Tomorrow